So thank you very much. So first of all, thank you, Alain, for organising this symposium and for inviting me. It's a real pleasure to be here. And thank you for allowing me to speak for up to an hour. Is that right? I'll try and set a good precedent. So what I'd like to talk about today is what I think of as the 4 Ps of Morphogens. So pattern, position, persistence, and precision. And the new work, the unpublished work I'm going to talk about today is from 2 students in the lab, Catherine Exelby and Edgar Herrera Delgado. And it's in collaboration with Peter Solich, who's a mathematician at King's College, London. So I don't think I need to really say anything to this slide, given the audience and given what Alain has already introduced us to. But just to remind you, sort of the textbook conventional view of what a morphogen is this idea that it's a signal secreted, produced from a local source within a developing tissue that spreads away from that source to form some kind of gradient. And then cells respond to that signal to adopt different gene expression patterns which ultimately determine cell type identity. And as Alain has already said, this has always been somewhat contentious and controversial. And many of you will be familiar with this paper from almost 50 years ago now, from Francis Crick. I thought it'd be relevant to mention this since I'm now at the Francis Crick Institute. But in this short paper, Francis Crick, essentially with back-of-the-envelope calculations, made an argument that diffusion could be a mechanism to explain pattern formation in embryonic tissues. But already at this time, this was a controversial, a contentious issue. And you can tell that because this elicited a response from Elizabeth Ducat, who wrote to Nature with this letter. So she, earlier in her career, had worked with Conrad Waddington. I think it's just interesting just to read this. So a couple of things I would point out is that she already says that this idea of diffusion is a long discredited concept, even in 1970. And what she goes on to say is that if materials have diffused in and out of my experimental embryos, I've regarded it as a sign that I'm dying or dead. So I think it's clear that even in the 70s, the idea of diffusion as a long-range signal was contentious. And it's also interesting to note that 40 years ago, there was still scientists were still robustly criticizing one another's work in public. Anyway, so partly for this reason, what I focused on and what I'd like to talk today about is not so much the extracellular mechanisms of morphogen signaling, but think about this from the perspective of the responding cell. So what a cell is doing is receiving some input from the morphogen, and it needs to convert that into an output. So gene expression will ultimately underlies cell identity. So what is the mechanism of that information processing? And to make this concrete, I'm going to talk about this in the context of the developing neural tube. So in the adult nervous system, and in particular in the spinal cord, which we focus on, different neuronal subtypes are spatially restricted. So in the dorsal half of the neural tube are neurons which process sensory information, sending it up into the brain or down into ventral regions of the neural tube, where motor neurons and interneurons that control motor output are located. So that organization of different neuronal subtypes arises early in development with the spatially segregated generation of those different cell types. So along the dorsal ventral axis, we can identify several classes of molecularly and physiologically distinct neuronal subtypes. And these arise from proliferating progenitors, which are also arrayed along the dorsal ventral axis. And these domains are molecularly distinct, and we can recognize them by the combination of genes expressed in each of these domains. And this pattern of gene expression is in turn dependent on signals emanating from the ventral and dorsal poles of the neural tube. And I'm going to focus on the ventral signal, sonic hedgehog, today. So in the absence of sonic hedgehog, the pattern of gene expression in the ventral neural tube is absent. So hedgehog signaling is necessary to establish this pattern of gene expression. Moreover, a range of different types of experiments suggest that hedgehog can function in a long-range manner in some kind of concentration-dependent way. So, for example, in this experiment, we're taking little regions of a developing chick embryo, so from the open neural plate before the neural tube is formed, and culturing these in vivo in ex vivo in defined conditions. So if we just take one of those explants and culture them without any added recombinant hedgehog, then the majority of cells in those explants express genes characteristic of the dorsal neural tube, like PAX7. A moderate concentration of hedgehog, so 1 nanomolar hedgehog, represses PAX7, and instead, the majority of cells express a red gene, OLIG2, characteristic of the middle of the ventral neural tube. Whereas 4 nanomolar, a higher concentration of hedgehog, instead of red gene, you see the green gene, NKX22, which is characteristic of cells right adjacent to the source of hedgehog at the ventral pole of the neural tube. So this looks like a classic kind of Walpertian concentration-dependent response, a morphogen. But of course, things are more complicated than that. So in addition to the concentration, the duration of signal is also influential. So if we take the same type of explants but look at those exposed to hedgehog signaling for different periods of time. So we take that high concentration of hedgehog, 4 nanomolar. I showed you the effect at 24 hours was to induce a high response gene, NKX22. But 12 hours after exposure to that high concentration, you see the low response gene, OLIG2. And if you quantify this, you see OLIG2 being induced prior to NKX22, but the expression of OLIG2 is transient. It goes down as NKX22 is being induced. And you see a similar sequence of gene expression if we look in vivo in the neural tube. So you see these sort of kinematic waves of gene expression moving across the neural tube, with cells expressing the low response gene before inducing the higher response gene. So we'd like to understand how both the concentration and the duration of hedgehog signaling are affecting gene expression. And importantly, we need to understand how you generate this pattern, so how you convert, so essentially the analog to digital conversion here, so how you convert the gradient into discrete switches in gene expression. And that's what I'm going to call the first P, pattern. And in fact, Hans Spemann made this point 80 or so years ago in an essay he wrote about pattern formation and the idea that gradients could be involved in pattern formation. He said the gradient must be conceived as continuous, whereas the series of formations whose differentiations will be determined by that gradient is absolutely discontinuous. So how do you go from a continuous gradient into discrete switches in gene expression? And I think insight into this has come from realizing that these patterns of gene expression are not only nice patterns that allow us to identify different dorsal ventral positions, but they also, these genes are also transcription factors, and they're linked together in a transcriptional network. And it's that transcriptional network which generates pattern, which generates the discrete switches in gene expression. And evidence for this initially arose from developmental genetics. So, for example, just to give you one example of this, these two transcription factors, PAX6 and NKX22, are normally mutually exclusive with a boundary between these domains in the ventral neural tube. In a PAX6 mutant, PAX6 protein is obviously gone, and what you see is a marked expansion in the domain of NKX22. So now NKX22 is expanded dorsally. So these type of experiments begin to allow us to put together a transcriptional network which connects those transcription factors together. And if we start to abstract circuits from those experiments, so you can turn this sort of wiring diagram into little cartoon circuits, what one notices is that it's dominated by cross-repressive interactions, so mutually exclusive, mutually repressive interactions between pairs of transcription factors. And one feature of those kind of interactions is that they form toggle switches. You mutually exclusively express one or the other, but you can't express both of those. So they provide a mechanism to generate discrete switches in response to a continuous input. But those experiments, those genetic experiments also suggest that not only is it important for pattern formation, for generating discrete switches, this network is also important for providing position in the neural tube. You change the domain of expression when you manipulate this transcriptional network. And to illustrate that, I'm just going to focus on this subnetwork containing 4 transcription factors, 2 of which, as you've already seen, NKX22 and OLIG2 are dependent on Hedgehog signaling for their expression. So this combination of 4 transcription factors defines 2 boundaries of gene expression within the neural tube, one between NKX22 and OLIG2 and PAX6, and the dorsal boundary of OLIG2, which corresponds to the ventral limit of another transcription factor, IRAQUA3. So the combination of those 4 transcription factors define 2 boundaries within the neural tube. And we can convert this cartoon of a transcriptional network into a dynamical systems model, so a model of linked ordinary differential equations. I think it's too early in the morning to go into all of the details of the equation, you'll be pleased to know. But actually, if you can spot the mistake on here, I'll buy you a beer later. Okay, so we can convert this cartoon into linked differential equations, and then we can start simulating this in silico. And if we do that, we can see it quite naturally generates that pattern of gene expression we're seeing. So if we simulate a gradient of Hedgehog signaling and then look at what response this network gives, we see a stripe of NKX22 activity, levels where Hedgehog signaling is high, stripe of OLIG2 at moderate levels of signaling, and then PAX6 and IRAQUA3 at low levels of signaling. So you can see these 2 boundaries of gene expression recapitulated by this model. Moreover, if we do the equivalent of those knockout experiments in silico, so we can remove an equation. So for example, we can remove the PAX6 equation from this, and what you can notice is the effect of that is to expand NKX22 activity to lower levels of signal, reduce OLIG2 expression, and the dorsal boundary of OLIG2 remains similar. And those in silico changes are reminiscent, recapitulate what we're seeing genetically. So in the absence of PAX6, we see an expansion of NKX22 expression. So this is all very reassuring, but this is actually how we establish the model. So the model can recapitulate what we're seeing. But what was more striking is that not only does it recapitulate those spatial patterns of gene expression, what we noticed is that the in silico model also generated the same dynamics of gene expression as we're seeing in vivo. So if we look at the dynamics of gene expression in response to a level of signaling that eventually results in NKX22 activation, we see that always generates a transient burst of OLIG2 expression. So this circuit gives us both the dynamics of gene expression as well as the spatial pattern. We can't separate those 2 things. And as you remember from those in vitro and in vivo experiments, we're seeing that same sequence of events. We're always seeing a transient burst of OLIG2 expression before NKX22 induction. So that transcriptional network gives us both the spatial and temporal sequence of events and actually links them together. It's in fact that it's a regulatory logic. It's the structure of that network that gives us both of those behaviors. And perhaps it's easiest to think about in one of these sort of cartoons of the what one could think of as the phase space of this network, where we're looking at the behavior of the network over time in response to different levels of Hedgehog signaling. So what you can see is in order to get NKX22, the high response, you need higher levels of signal than for the induction of the low response gene OLIG2. But you also need that signal for a longer period of time to get NKX22 induction than to get OLIG2 induction. So this transcriptional network links both the temporal and spatial pattern together. So another way of looking at this is to plot the behavior of gene expression in gene expression space. So you're starting at a PAC6 high. Yeah, I should explain. This is level of PAC6, low to high, level of OLIG2, level of NKX22, low to high. So you're starting in a PAC6 high, NKX22 low state, and you're adding sufficient signal to eventually induce NKX22. But the system, rather than moving directly from the PAC6 high to the NKX22 high, has a transient diversion towards the OLIG2 high state before returning to the eventual steady state of NKX22 high, OLIG2 PAC6 low. And that behavior in terms of dynamical systems nomenclature is called a dynamical ghost. The system is actually moving towards a steady state that doesn't exist. There's a ghost of a steady state of an OLIG2 there before it relaxes to the actual steady state of the system, NKX22. And that'll become important in a couple of slides' time. So together that suggests that both the pattern and position of the morphogen is determined by the transcriptional network. This transcriptional network takes the morphogen input and converts a continuous input of morphogen into discrete switches in gene expression and positions them at appropriate locations within the neural tube. So far, I've implied that the level of Hedgehog signaling is constant throughout this, and the simulations are all done with constant levels of signal. But reality is more complicated than that. And we got a hint of this when we started to look at the dynamics of Hedgehog signaling within the neural tube. So using just a very simple reporter for Hedgehog signaling, so the Gli binding sites, the transcriptional effector of Hedgehog signaling, linked to fluorescent reporter and making transgenic animals with these. So you can see this reporter is active in regions of the embryo where Hedgehog signaling is active. And within the neural tube, we can now look at the dynamics of signal, dynamics of glia activity over time, just by measuring the level of fluorescence from ventral to dorsal in the neural tube. So if we do that, if we measure Hedgehog signaling over the developmental time window that pattern is being established from ventral to dorsal in the neural tube, what we can see is each of these developmental times you see a gradient of glia activity, high ventral to low dorsal, but the amplitude of that gradient changes quite markedly over time. So initially it's quite a low amplitude gradient, rapidly reaches a peak amplitude before relaxing back down to low levels. So that means from the perspective of an individual cell within the neural tube, it's not seeing a constant level of glia activity. That level of glia activity is changing over developmental time. Moreover, the same dynamics can be seen when we challenge explants with a constant concentration of recombinant hedgehog. So here we're using the same reporter in those explants, and what you can see from these assays is those same dynamics. You rapidly reach peak levels of glia activity before they decrease. So somehow the cells must be converting those dynamics of gene expression into the appropriate patterns of gene expression and then maintaining those patterns of gene expression even as the signal decreases. And in fact, looking back at the transcriptional network, a potential mechanism for this immediately suggests itself. And that's this idea of hysteresis, which if you've looked at dynamical systems and looked at bistable switches, you wouldn't be surprised by. But that idea of hysteresis is perhaps best explained if we just think about the expression of the high response gene NKX22. So what I've shown you is that NKX22 requires high levels of glia activity, high levels of Hedgehog signaling to induce expression. And that's because if you start with the absence of Hedgehog signaling, repressors of NKX2-2 are being expressed. So to overcome all of this repression, you need a lot of Hedgehog signaling for NKX2-2 to be induced. But once induced, NKX2-2 is able to repress its repressors, and therefore you need less Hedgehog signaling to maintain NKX2-2 once it's induced. And that gives rise to this region of bistability and this phenomenon called hysteresis, where once induced, NKX2-2 is easier to maintain. So you can reduce the level of Hedgehog signaling and still maintain NKX2-2 expression. So this provides a putative mechanism to explain how gene expression is maintained even after this level of glia activity, level of Hedgehog signaling is decreased. And this prompted for us to do an experiment to test this idea. Again, taking advantage of that ex vivo, those explants, to test this. We could take explants out of a chick embryo and culture them in high concentrations, 4 nanomolar hedgehog initially for 18 hours. And that induces the high response gene NKX2-2. If we continue exposure to that high concentration, we maintain NKX2-2 expression. If at 18 hours after we've induced NKX2-2, we reduce the level of Hedgehog signaling to about a third of the initial level of signaling, we still maintain NKX2-2. But if we use that lower level of signal from time 0, we get very little, if any, induction of NKX2-2. So that's consistent with this idea of hysteresis. Once we've induced NKX2-2, we can maintain it with lower levels of signal than is initially necessary to induce NKX2-2. So taken together then, that suggests that this transcriptional network provides 3 features of pattern formation. It takes an input of Hedgehog signaling, generates pattern by converting the continuous input into discrete switches in gene expression, positions those boundaries within the neural tube, and also maintains those patterns of gene expression even after the levels of signaling decline. So that's the first 3 Ps. I've given you pattern, position, and persistence. So in the last 5 minutes or so, I just want to talk about some new work which looks at precision. And this really comes from noticing when we looked at mutants is not only are the boundaries of gene expression, the position of gene expression changed, but also the precision of gene expression is altered. So if we look at NKX2-2 expression in a PAX6 mutant, we see the domain of gene expression is expanded, but also the boundary of gene expression is no longer as sharply defined. And you can see intermixing between OLIG2 expressing cells and NKX2-2 expressing cells. So we hypothesized this could be a consequence of noise in gene expression that arise from the finite molecular copy numbers of the proteins within cells. So to test this, we can convert the deterministic differential equation model that I've introduced you to a stochastic differential equation by adding a noise term based on a chemical Langevin equation formalism. And this then describes the effect of molecular fluctuations, the effect of stochasticity on gene expression. So if we do that for the wild type circuit, we can see that this results in decreased some noise in the precision of gene expression between NKX2-2 and OLIG2 in the simulations. What was surprising is that just by adding the noise, this recapitulated the increased imprecision we're seeing in the PAX6 mutant. So the same model, just removing the PAX6 equation from the model, resulted in both the expansion of the domain of NKX2-2 and the decreased precision in which that new boundary it now forms. So we can get maybe a better intuition to this if we look at this from the perspective of gene expression space. So if we think about this, so again, you're looking at 3-dimensional gene expression space with PAX6, OLIG2, and NKX2-2. So NKX2-2, the high response gene, the high response is here, and the starting position is here. So if you look at the consequences of noise, to go from a PAX6 high state, OLIG2 high state, to an NKX22 high state through molecular fluctuations, through the effect of noise, you have to transition through this critical point here, this saddle node within the system. And what you can see is when we look at the fluctuations introduced by those stochasticity, those fluctuations result in increased variation in gene expression, but they're angled slightly away from the critical point, so they're not directly towards the critical point. So that makes fluctuations in gene expression, it makes it more difficult for the system to transition from an OLIG2 state to an NKX22 state just by stochasticity. However, if we think about this now in a PAX6 mutant, we obviously remove the PAX6 dimension, so you flatten this all down onto a two-dimensional dynamical surface. And now the fluctuations are directed much more towards that critical point. So it's much easier for molecular fluctuations to result in a cell reaching that transition point and therefore noise driving it into an NKX22 state. So this raises the question of whether other perturbations to the system could also affect boundary precision. And just bear that in mind, because this becomes relevant when I show you the next set of experiments. And this arises because we became interested in enhancers, so the cis-regulatory elements which control key genes. And in particular, if we look at the OLIG2 gene, there's a putative cis-regulatory element, 33 kb upstream of that element, so it's accessible by ATAC-seq, and it also binds key transcription factors including NKX22, Hedgehog effectors, the Glee proteins, and also SOX2, which is a pan-neural activator of neural genes, so it results in the basal expression of OLIG2. Moreover, Tony Eustavine has shown when you take that element in isolation, it's sufficient to drive OLIG2-like expression within the neural tube. So what Catherine Exelby in the lab did is she generated mice where that element has been deleted, so using CRISPR to specifically delete out that enhancer element. And what you can see comparing wild-type animals to mutant animals in which that element is missing. So OLIG2 is still expressed, but you can see that the domain of OLIG2 has changed. And there are three obvious changes. So first of all, the dorsal limit of OLIG2 is shifted closer to the ventral neural tube, so there's a smaller domain of OLIG2 in this mutant. The level of OLIG2 expression within the progenitor domain is somewhat decreased. But what was quite striking is that this boundary with the NKX22 domain, although it's in approximately the same position, it now looks much less sharp, much less precise. So you see intermixing of cells between the NKX22 and OLIG2 domain, which is much rarer within the wild type. And that's more obvious when we quantified this. So quantifying in multiple embryos the level of PAX6, OLIG2, and NKX22 in individual cells, you see at the tissue level this restriction, so this decrease in the size of the OLIG2 domain, so a shift from more dorsal to more ventral in the neural tube, but the ventral boundary appears to be relatively well conserved. But when we look using several statistics here, just looking at the neighbors of individual genes and individual cells and looking whether they're expressing the same or different genes, you see an increase in imprecision within the enhancer mutant compared to wild type. So there's increased mixing between the OLIG2 and the NKX22 domain within the neural tube. So we wondered whether this could be recapitulated with the in silico simulations. So as I mentioned from those genomic data, that regulatory element has several binding sites in it, including binding sites for NKX22 and the Glee activity, Glee transcription factors, the Hedgehog signaling activity, and also binding sites for SOX2, a pan-neural transcription factor. And what Edgar found is that when he reduced the parameters which describe those interactions on OLIG2 within the model, he could recapitulate all of those phenotypes. So what you see is the single parameter change, single set of parameter changes, both restricts the dorsal limit of gene expression, reduces the level of OLIG2 within that domain, and results in an increase in imprecision at the boundary between NKX22 and OLIG2. So we again can now look at this from the perspective of gene expression space and look at the effect of the direction of fluctuations on those alterations within the model. So now looking at, in the wild type, looking at the fluctuations as we move dorsal in the neural tube. So you can see when we're close to the boundary, the fluctuations are quite close to this critical point, but because they're angled away from that critical point, as you move dorsally in the neural tube, then fluctuations very rarely result in gene expression reaching this transition point. By contrast, if we look at the same simulations in the enhancer mutant conditions, what we see is that that angle of the fluctuations is angled more towards that transition point. So it's easier for fluctuations in gene expression to reach the transition point and therefore cross over and into an NKX22 expression by stochastic mechanisms. So taken together then, what I wanted to emphasize is this idea that the transcriptional network is crucial for understanding how the cells are interpreting hedgehog signaling. So I've argued it provides a mechanism for generating patterns for discrete switches in gene expression. It positions those boundaries within the tissue. It provides a mechanism for persistence, so for maintaining gene expression once it's been established. And the structure of the network also provides precision to those decisions as well, increasing the sharpness of boundaries within the neural tube. So I'm going to leave it there. I want to thank the people in my lab that have been responsible for this work. In particular, I want to acknowledge Catherine Exelby, Edgar Herrera Delgado, and Peter Solich, the new work, the unpublished work that I presented today. And I thought it would be an appropriate opportunity, particularly with the current editor-in-chief in the audience, just to mention development one more time. I mean, I don't need to emphasize this to you in this audience, but development is a journal you should be submitting your papers to, and I look forward to handling them all in a few months' time. So thank you very much for your attention.